Hello guys, hello friends, hello G's, it's your boy again, falling with a star. So guys, I want us to talk about smooth muscles. And these smooth muscles, they are being found in hollow organs inside the body, like the GIT, the stomach, the blood vessels. So let's look at this structure here. This is the structure for the smooth muscle. And looking at the structure of the smooth muscle, you can see that it is spindle shaped. It is spindle shaped. And the smooth muscles, they are uninucleated. They have only one nucleus at the center. So let's look at this structure here. But before we talk about this structure, let's look at the structure of what the skeletal muscle fiber. We said that in the skeletal muscle fiber, we have these myofilaments, and we have two types, the thin and the thick myofilaments. And we said that the thin filament, they are being attached to these Z-discs by a protein we call titin. But when you come to the smooth muscle, this is the thin filament. And these thin filaments, they are not being attached to what Z-discs, but rather they're being attached to this deep circles here. These deep circles, they are called the dense bodies. So the titans are rather the actin, or the thin filaments are rather being attached to these dense bodies. And these dense bodies contains a protein we call the alpha actinin. They are being made up of this protein alpha actinin. And you can see that these dense bodies are connected to each other by the structure we call the intermediate filament, which is also being made up of a protein, two proteins we call the desmin and the vimentin. Looking at the surface of the smooth muscle, you can see that it has these invaginations, and these invaginations are called the cavuli. It is similar to the T-tubules in the skeletal muscle. The cavuli, this is their function. What they do is that they conduct these electrical impulses, or these impulses flowing on the surface of the smooth muscle into the cell. You understand? Into the cell, and they are rich in voltage gated calcium channels. Calcium channels. So when they receive these impulses, they cause these voltage gated calcium channels to open for calcium to flow in, causing depolarization. Depolarization in the smooth muscle is mainly by calcium ions, not sodium ions. In the smooth muscle, you see, in the skeletal muscles, depolarization is by the inflow of sodium ions, but in the smooth muscles, the depolarization is by the influx of calcium ions. You understand? So let's look at something here. Let's take the myofilaments here. The myofilaments, I said that we have the thick and the thin myofilament. So let's pick the thin myofilament. You see, the thin myofilament is being made up of two proteins called the tropomyosin and the actin. But in the skeletal muscle, we said that the thin filament is being made up of three protein components, which is the tropomyosin, the actin, and the troponin. So in the smooth muscle, there is no troponin. There is no troponin. The thin filament is being composed by these two, actin and tropomyosin. There is no troponin, but rather they have this protein we call cammodulin. The cammodulin. And in the skeletal muscle, what did we say what did we say is the function of the troponin? We said that the troponin has three subunits: the troponin C, the troponin I, and the troponin T. The troponin C is the site where calcium binds to, and when calcium binds to that site, it causes a conformational change in the troponin. And the troponin is being attached to the tropomyosin. So a conformational change in the troponin also affects the tropomyosin to also undergo a conformational change. You understand? And that will expose the myosin binding site on the actin. But in the smooth muscle, we have this cammodulin, and that are the places where the calcium that are flowing inside or that are flowing into the cell will bind to. So the cammodulin, they have this calcium binding site. So the calcium will bind to that part, and they form calcium cammodulin complex. They form a calcium cammodulin complex. And what does this calcium cammodulin complex do? What they do is that this calcium cammodulin complex will go and activate this desmon. go and activate this cardesmon. And what is this cardesmon? This cardesmon is being attached to what? The tropomyosin. And this cardesmon controls or regulates the tropomyosin. It can tell the tropomyosin to block the what? The myosin binding site on the actin, or it can tell the tropomyosin to what? Expose the myosin binding site on the what? On the actin. So the cardesmon, which is being attached to the tropomyosin, regulates what? The tropomyosin. So this calcium carmodulin complex will go and activate this cardesmon, and it will cause the tropomyosin to what? Expose the myosin binding site for the myosin hairs to bind at that site to cause what? Contraction. So this, let's say that this is the myosin, the thick filament, this is the head, and it binds to this myosin binding site on the what? On the actin, you understand? And the head of what? The myosin is called cross bridges. Let's look at something here. Let's look at the innervation of what? The smooth muscles. You see, when you take the skeletal muscles, they are being innervated or being stimulated by what? This somatic nervous system. The somatic nervous system, because the skeletal muscle is voluntary, can consciously be controlled. You understand? So they are being innervated by what? The somatic nervous system. And you see that this is the axon and this is the axon terminals. And the axon terminals, they contains these neurotransmitters which are in these vesicles. The neurotransmitters, they are in these vesicles. You understand? So the axon terminal, and the neurotransmitters in these vesicles. The neurotransmitters, they carry what? Action potentials, and upon their release, they bind to receptors on the surface of what? The skeletal muscle fibers, and initiate what? An action potential in the what? In the skeletal muscle fiber, which will lead to what? Contraction. But let's look at the smooth muscle. The smooth muscle, this is the what? The axon. And on the axon, we have these circle-like structures called the varicosity. These circular structures, they are called what? The varicosities. And the varicosities, they contain the neurotransmitters. And these varicosities, they are the ones that will release these neurotransmitters. And these neurotransmitters will bind to what? The receptors on the smooth muscles, and causes their what? Their contraction. You understand? And causes their contraction. You see, these smooth muscles, they can be innervated. By these neurotransmitters, the acetylcholine, the non-epinephrine, the glutamate, you understand? But when you take the skeletal muscles, the neurotransmitters that actually stimulates these skeletal muscles is the what? Is the acetylcholine. Only acetylcholine. But in the smooth muscles, we have the acetylcholine, the non-epinephrine, and we have the glutamate. So these neurotransmitters will bind to the receptor on the what? On the smooth muscles, and they initiate what? An action potential here, depolarization will occur, which will lead to what? The contraction. You understand? The contraction. Now let's talk about the myosin head. When you look at the myosin head actually have these myosin light chains. The myosin light chains. And these myosin light chains, it can exist in two states. So let's pick the states. The state one, when these myosin light chains are have what? Phosphate being attached to them, they become activated. But when they don't have this phosphate attached to them, they become inactivated. You understand? So when phosphate is being attached to this myosin light chain, they become activated. When there is no phosphate, they become what? Inactivated. And they become what? Inactivated. And these phosphates are being added to this myosin light chain by a certain protein, a certain enzyme called the myosin light chain kinase. Myosin light chain kinase. You see, kinase is they phosphorylate things. So this myosin light chain kinase is the one going to put the phosphate on the myosin light chain. They will put the phosphate on the myosin light chain to activate it. Understand? And we have another enzyme which we call the phosphatase. So the myosin light chain phosphatase, it actually removes what? The phosphate from what? The myosin light chain, and causes it to be what? Inactivated. You understand? Inactivated. And when these myosin light chains are in their activated state, it causes the myosin hair to attach to what? The myosin binding site. And the myosin binding site, and initiate what? Contraction. And initiate what? Contraction. Let's look at some facts about what skeletal, some facts about smooth muscles, some facts about smooth muscles. Smooth muscles, they don't have what? A sarcomere. You see, sarcomere is from one Z days to another Z days constitute what? The sarcomere. And this is the case that in the smooth muscles, they don't have Z days. So there is no what? Sarcomere. There's no striations, because in the skeletal muscles, it is due to the arrangement of what? The thick and the thin filaments that gives them the striation characteristic. But here, they are not being arranged in that manner, so there is no striations. They don't have branches. In skeletal muscles too, there's no branches. They don't branch. But in the cardiac muscles, they branch. You understand? They branch. They are not voluntary. They are not voluntary, so they are involuntary. They are unconsciously being controlled. They are unconsciously being controlled. They don't have troponin, as I said. Instead of troponin, they have what? The cammodulin. That's where the calcium will bind to, and they form the calcium cammodulin complex, and which will what? Activate the caldesmon. And this caldesmon will tell the tropomyosin to expose the myosin binding site for the myosin here to bind to that site to initiate what? Contraction. They have undeveloped sarcoplasmic reticulum. They also have thin filaments. Their thin filaments are connected to what? Dense bodies, not Z-days, dense bodies. Depolarization is by what? Calcium channels. Depolarization is by the influx of what? Calcium ions, not sodium ions. The main source of calcium ions is not the sarcoplasmic reticulum, but rather the extracellular fluid. And the smooth muscles, they use what? Less ATP. So this is the last part. Let's talk about the types of what? Smooth muscles. The types of what? Smooth muscles. You see, we have two types of smooth muscles. We have the unitary, which can also be called the visceral, which can also be called the single unit smooth muscle. The unitary or the visceral or the single unit smooth muscle. And we also have the multi-unit smooth muscle. You see, before I even talk about these types, let's talk about the gap junction here. The gap junction is also known as the nasos or the nesis. So this is the smooth muscle, and all of them are being linked by this gap junction. And what this gap junction does is that when one of the smooth muscles are being stimulated or is being stimulated, the action potential generated in this smooth muscle will be what? Conducted to what? These smooth muscles will be conducted to these smooth muscles, which actually leads to the contraction of all of them together. So all of them will contract simultaneously. Because of this gap junction, the action potential, let me repeat myself again. Because of this gap junction here, the action potential being generated here will be conducted to this smooth muscle, and that will cause them to simultaneously what? Contract. And we call that syncytium or the syncytium. When different muscle cells are contracting together simultaneously, we call that the syncytium. So this gap junction helps in that syncytium. So now let's move to the types. We have the unitary. And for the unitary, they have these gap junctions. The smooth muscles, they are being linked by these gap junctions. So when one of these smooth muscles are being stimulated, the action potential generated in it can be conducted to the other smooth muscle, and together they all contract simultaneously. So for the unitary, the smooth muscles, they contract simultaneously as one unit. You understand? And they can be controlled by neural pathways. That is the nerve cell stimulating them, and in front of the neural pathway. And they can be stimulated by hormone, the endocrine pathway, and they can be stimulated by metabolites and also drugs. And these unitary smooth muscles, they can be found in the GIT and also in the blood vessels. Let's look at the multi-unit. For the multi-unit, the muscle cells in there, they don't have gap junctions, so they are not being linked. So when one smooth muscle is being stimulated, the action potential generated in that smooth muscle cannot be conducted to the other smooth muscle, so they contract independently. All of them needs what their nerve endings to stimulate them for them to contract, so they contract independently, and they are being controlled, or they can be stimulated by only by what neural pathways, only by nerves or the autonomic nerves. You understand? So they can be found in the ciliary muscles of the eye, the iris, and also when you take the penis, they can be found in the vas deferens. So that's it for smooth muscles.